Nice to meet you. My name is Emanuelle Abbagnano, and I'm a PhD here at Imperial with Dario. And today I'll be talking a little bit about my PhD project here. So we'll be talking about non-invasive peripheral neural interfacing for model augmentation, which is something that we have covered quite extensively during these days. So, and what is model augmentation? So whenever we aim to extend like a person's capabilities, movement abilities, we're looking at motor augmentation, as there's different examples of this. And not only different examples, but different types of augmentation. We can do power augmentation if we want to augment the power, the force of a person. We can do workspace augmentation if we aim to extend the volume in which a person can work, or degrees of freedom augmentation. Now, in all these cases, what we need is a control signal. Theoretically, the best would be a biosignal, so something we can extract from the body, and is there for us to be used for controlling external degrees of freedom. If we want to do a peripheral interface, so let's say that we apply a grid, high-density EMG grid or whatever, EMG, the peripheral muscle that we activate to extract neural information being transmitted from the spinal cord, we can get two main inputs to the muscle that we can possibly use, maybe two types of information. That is the effective bandwidth below 10 Hertz and the beta band between 15 and 30 Hertz. Now, the effective bandwidth is being used from the body to control force. Theoretically, we will expect to use something that, ideally, that it's free, it's there, and that is not used for anything else. Beta band specifically doesn't have necessarily a role at peripheral level. We haven't found so far what it is doing. We know for sure that it's not controlling force in any way, directly or indirectly. So it could be a good candidate for motor augmentation as a control signal. Now, where does it originate from? Is it why is it peripherally? Well, theoretically, probably it originates from the motor cortex, and if we look at this, cortical beta band, so this oscillation between 15 and 30 Hz are associated to sensory motor control. We don't know, though, the functional role of these oscillations at cortical level yet. We just know that they are associated to sensory motor control, and that whenever there is an asymptotic contraction, that the cortical beta band power is increased. All right, so it's increased, and it's phase coupled to muscle. So it means that we find the cortical level, and somehow it's coupled to peripheral level when there is an asymptotic contraction. So this likely means that these cortical oscillations are being projected to peripheral active muscles. And there is different studies that have shown this coupling, and they've studied this transmission of this cortical oscillation to the periphery. Now, so let's go back to the periphery. So let's say that we extract these oscillations from the periphery. And we do know that they can be decoupled from muscle contraction. So we're doing asymptotic contraction, we're extracting these oscillations, and that we can voluntarily modulate them when we are doing when we give feedback to a person of these oscillations. There is also some cons, though. For example, its control is neither easy nor intuitive, which is something that we need to tackle. If we go and look at the time series of the signal, it looks as a chaotic signal. It's not easy to control in order to get the hang of it. And importantly, we don't know if peripheral beta band to a muscle is uniform. We extract it at some contraction level, but is the beta band that we are extracting at that contraction level the same that we extract at another contraction level? Meaning that is the beta band project uniformly to the entire motor neuron pool of a muscle? So the final question is, can we learn to reliably and efficiently control beta band activity peripherally for motor augmentation? To answer this question, the first thing that we need to address is the last point, which is we don't know if this peripheral beta band is projected uniformly to the muscle. To answer this question, we made an experiment where we designed an experiment with 12 healthy volunteers, where basically we are applying both EEG and high-density EMG from the tibialis anterior muscle, and subjects are doing this ankle dorsiflexion at different contraction levels. We are applying both EEG and high-density EMG, and to analyze the neural dynamics of this transmission from cortex to muscle. Now, the most important thing is that we want to see the projection of these oscillations across the entire motor neuron pool of the muscle. So as you are increasing the force, we expect to find different size motor neurons. And so we are doing 6 different types of asymptotic contraction, from 5% mild contractions to submaximal contraction level, 70% MDC, while we are recording EEG and high-density EEG. And we expect that, because of the technical limitations of the decomposition algorithm, will be decomposing different size motor neurons and effectively then analyze how this projection occurs of this oscillation from cortex to muscle. For the EEG, we bandpass filter the data. We apply independent component analysis from some artifacts. We apply the surface interpolation, and that gives us the processed EEG, particularly using the CZ channel. And for the EMG, instead, we bandpass filtered with the motor unit decomposition to get the common spectrum. And this common input normalization, which if you want, you can learn more about directly on the paper that is online. So when we get the processed EEG and the cumulative spectrum, what we are going to analyze is the neural dynamics between these two signals to characterize beta and its transmission. One thing that we'll be looking at, for example, is the coherence, which is a measure linear correlation between these two signals as a function of the frequency. So basically, we're just looking at how correlated and how similarly these two signals fluctuate between cortex and muscle. And so, for example, if you're doing cortical muscular coherence, you're applying this measure to the cumulative spectrum and the EEG. If you're doing intramuscular coherence, you're just doing it between two independent pools within the same muscle of the motor neurons. We're also looking at cumulative density, which is basically just a way for us to understand how long it takes for this cortical oscillation to be transmitted to the periphery in milliseconds. So let's look at the results. So first thing first, we got on the x-axis all the 6 different contraction levels we acquired during this experiment. We can see in the y-axis that the recruitment threshold of the decomposed motor neurons at each contraction level is increasing. Effectively, the recruitment threshold is an index of the size of the motor neuron. So we did decompose different size motor neurons across these different contraction levels. So let's see now the neural dynamics for these different motor neurons. Oh, sorry, this is just the number of motor neurons decomposed at the contraction level. If you look at the cortical muscular coherence on the left and the intramuscular coherence on the right, there is no significant changes for these two measures. So dashed line is the significance threshold, so everything above is above noise level. Right axis, again, the recruitment threshold that we've just seen. So we're looking at differences motor neurons, and we can see that even though these motor neurons are different size, the coherence is not changing across these different groups, meaning that probably the information being shared and the common input is not changing. It's this beta band is projecting uniformly to different size motor neurons. Also, if you look at the power of across these different size motor neurons, both at cortical level and peripheral level, on the left, there is no change in the power spectral density in the beta band. So coherence in terms of the frequency spectrum, the information that's being projected to the motor neurons is not changing, neither is the power of this information. If you look at the common density, so if you look at the delay of this information in the beta band being transmitted from the cortex to the muscle, there is a decrease. So for small motor neurons, there is a higher delay, for bigger, smaller delay. We know that because bigger size motor neuron will have higher conduction velocity. So if we account for the different peripheral conduction velocity, so we take the conduction time of the cortical spinal tract, which is 13.9 milliseconds. We subtract from those delays this time, and we take into account an average common peroneal nerve, so from the spinal cord to the tibialis anterior muscle delay, which is length, which is 500 millimeters, and we calculate the peripheral conduction velocity for two different groups of motor neurons at 5% and 70%. We get two different conduction velocity, which are within literature values. So effectively, we can account for this reduction just by taking into account the different conduction velocities of peripheral motor neurons, which also tells us that it's also always the cortical spinal tract that transmit this same beta band. And so we know that cortical beta band is transmitted through the cortical spinal tract to the motor neuron pool, and then once it gets there, it's transmitted uniformly to the entire motor neuron pool. So key points. Well, we do know now that cortical beta projects uniformly to the motor neuron pool independently of the size of the motor neurons transmitting it. That this delay is in accordance to being the cortical spinal tract transmitting beta band always at any contraction level, and that this beta band is projected across the whole motor neuron pool with constant power. Great. So we got a reliable signal that we can extract at any contraction level for motor augmentation, which is this message. So now we need to address the other 2 points, which is that its control is not easy or intuitive. We need to extract this signal and make it intuitive, easy to control. So we can get something from the literature. So we already heard from the study during the summer school And so that beta band is involved in this cancellation mechanism, which is whenever we cancel a movement, there is an increase in power at cortical level. And this showed that whenever we look at the peripheral level, this cancellation, this increase in power in the beta band is also transmitted out of the peripherally active muscle, which is what we are seeing right here. So theoretically, we can look at it in this way. We can look at this as a true state signal that we can use for motor augmentation. And so whenever the person wants to change the state of what is controlling, it just needs to sense beta band high by canceling a movement. Now that we know that we have a simple signal to use, maybe, we need the person to learn to use it. And so for this, we designed an experiment, which is a 10 days training where the person are learning to control the signal through this cancellation mechanism over time. We have a control session, a final session at the beginning and at the end of the training to see if there is a learning, if there is also neuroplasticity associated to it. And so these control and final sessions are made with both EEG and high density EEG. And what you're seeing the subject doing is just different isometric construction. So it will be 10% NBC, 20% NBC. The 10 days training instead, we built an interface that will help the subjects to train. And they're doing with all we're using is an ankle dynamometer and bipolar EEG. So we're extracting beta band from the muscle non-invisively with bipolar EEG. And then we're giving the feedback to the subject to it. And then he's engaged in different types of training to learn to control this cancellation mechanism, this signal for control. So how is the protocol for the training? So there is a different days. So every day we do the calibration to acquire different parameters for the training. Then there is this learning where they are exposed to what the cancellation mechanism is to learn how it works. And then there is a training where they're actively engaged to use it. As in any neurorehabilitation protocol, we begin with just 1 training sessions, and then we make it more difficult or make it more extensive by adding from day 5 another session. And then there's this test session the last 2 days. So again, the learning interface is this one. So this is where they're learning and they're being exposed to the mechanism and how it works. We just replicated what we already did. So there is the go, no go paradigm, which is what we're seeing here. So they will go at 10% NBC contraction level. Then from there, they will be ready to either no go, don't contract, or go. And this is what we know, so that they get exposed to this mechanism. Once they do this, right after, there will be training. So this is the interface. So what you're looking at is, again, they will be at 10% NBC with their first level. And then there will be this spike going from the right to the left inside the red area. Whenever it goes inside the red area, they're asked to cancel the movement and so increase the power in the beta band peripheral. If they're able to do so, we'll give them a well done. It's all gamified. So if we look at the results, we can see that across the 10 different, this is for subjects, across 10 different days, there is an increase in power in the beta band peripherally, whenever they're canceling the movement, which means that it becomes more obvious for us to see that they're actually canceling the movement and that the power in the beta band is actually increasing. And this is happening for all 4 subjects. So they are learning to use it, and it becomes more obvious, more intuitive, and more easy for us to spot that they're canceling the movement, and so they're changing the state of this controlled signal. If we're looking for some neuroplasticity associated to this change, we can see that for the power spectral density in the beta band, peripheral level, there is no change. But remember that we're looking at the signal that is of cortical origin, and there is an increase for 3 subjects at cortical level between the first session and the last session for the EEG. And if you look at the coupling between cortex and muscle, also there is an increase for all 4 subjects. And we think that these are indexes of some sort of neuroplasticity happening between cortex and muscle after being exposed to this training. So it is possible to improve voluntary modulation of peripheral beta band. And when we analyze it, we can see that it's associated to some sort of neuroplasticity. And with this, I'd like to thank you very much.